So just to summarize that specific genetic defect, it affects children and adults. It may have a long course, even many decades. And as I mentioned in the overall study from Dr. Steele, there are many different forms, UIP, CIP, and SIP. One of the most interesting parts about the family originally that I showed you by Dr. Thomas is that many of them seem to have triggering, especially the children. So they were apparently well until they had a respiratory viral infection that was proven much of the time, RSV, influenza, Asian flu in 3 of the individuals and in the family that I showed you first. So whether there's an interaction between respiratory viruses that many physicians have had a suspicion, certainly is possible, but is not yet validated, I think, with certainty. So surfactant protein C is an important cause, but it's a rare cause. It's only 1% of the families. So ABCA3 is another gene mutation that's been identified in newborns. So the strategy we're using basically is to use any information anywhere to try to learn which genes cause it. One of the most productive regions has been from Dr. Nogi and Whitsitt and Schulenine about diseases where surfactant is very important after you're first born. And so they have also investigated ABCA3. I'm not going to go through all of this, but it's another protein that helps in producing all of the surfactant proteins. And there may be many different genes and proteins in the pathway that make the proteins that come into the surfactant. And I mention it because it looks like combinations of different abnormalities or defects may feed on one another, and Dr. Nogi showed that in a report recently where 3 young babies had mutations in both. So the ABCA3 change, in order to get the overt disease from it, requires that you have copied variants on both of your ABCA3 alleles. It's a recessive gene by itself, so it's uncommon to cause disease because you have to have both of those. But when you have one ABCA3 allele and a surfactant protein C mutation, then that may be one explanation for why the disease begins earlier. So the neonatologists, including the group at Cincinnati and Hopkins, have made wonderful contributions that we're trying to mine their data and to see how that applies to adults with interstitial lung disease. So next I wanted to show you work that I think is really I was delighted to be a part of, but it was conceived by Mary Armanios in the center here with my colleagues at, so one of the most revered lung conferences is in Aspen, Colorado every year in June, and it really has turned out to be a spectacular, not only a spectacular place, but it's also a wonderful environment for a conference. But I'm going to show you work by Mary Armanios, and then William Lawson is in our group at the right. Mary is a oncologist at Hopkins. When she did the work I'm going to tell you about, she was actually a fellow in training with Carol Greider. Carol Greider is the Daniel Nathans Professor and Director of Molecular Biology and Genetics at Hopkins. She, 20 years ago, discovered telomerase in the labs of Elizabeth Blackburn. So it's amazing progress, and then application of understanding of that enzyme, protein enzyme, and the genes that control it are in part the story of IPF in families. So Mary Armanios, who I showed you momentarily, was working with Carol Greider. They were interested in the disease at the top of this list because telomerase mutations had been shown to cause dyskeratosis congenita. So there are a variety, these are all rare diseases, some more rare than others, but even very rare diseases, although they may not have much public health consequences, they have huge importance for understanding disease processes. And I believe that's the case for dyskeratosis congenita that I thought I'd tell you about briefly. So it's fascinating that it was discovered clinically, and these are obvious clinical findings that I'll show you some tables from the work of Dr. Dokow in London. But it was actually discovered, I think this was a Swedish article in 1910. So it's been known for a long, long time. The basis was never known until about 15 years ago when individuals, the Human Genome Project, allowed to understand what the molecular basis was for this disease. And although this is in Swiss, I think it tells the major manifestations, atrophy acutis reticularis. It's a reticular skin rash. Dystrophia unguium is the fingernails. So the fingernails are very poorly developed or absent. And then young individuals get leukoplakia, white plaques on their tongue that may turn out to be malignant or pre-malignant. So the disease had been known clinically for a long time. Here's from Dr. Dokow in 2000, some of the skin rash. I'm afraid it doesn't project very well, but you can see the missing or malformed nails and the leukoplakia in the bottom right panel. So this was a very poorly understood disease. The most tragic consequence of it is that teenagers develop bone marrow failure, so aplastic anemia or myelodysplastic syndrome develops, and this shows the probability of developing bone marrow failure by age and years. So you can see by age 20, 80% of the individuals have developed bone marrow failure in dyskeratosis congenitus. So what ties all of these kind of disparate, different manifestations together? Here's the percentage manifestations from Dr. Dokow showed you, the abnormal skin and nail dystrophy and leukoplakia. The bone marrow failure is almost universal, but at the very bottom part of this, the pulmonary disease, which ends up being IPF, is the part that's most important today. There's some other less common variants like extensive dental caries or esophageal stricture. Even though those are far less common, the concurrence of those, or osteoporosis, a very common problem, or urethral stricture, but also to mention the liver disease there that's only listed as 7%, but it ends up that those are very important as we learn more about extrapolations from dyskeratosis congenita. So the genes for dyskeratosis congenita are listed there. The first known was dyskerin, that's on the X chromosome. And then the telomerase genes, all of those genes affect telomerase function, and the TERT and TERC are the most important. Now there's, at the bottom, the others are extrapolations. So the telomeres are the ends of the chromosomes, and in fact, Elizabeth Blackburn, whose lab it was discovered in, describes them as the plastic tip around the tip of your shoelaces. It keeps them from becoming frazzled and fraying apart. So all the chromosomes have these repeat sequences, 6 base repeat sequences at the end of the chromosomes, and they are critical structures to protect the chromosomes. Here's a fluorescent staining where you can see the white telomeres on the end of each of the different chromosomes. And if those telomeres are lost, then the chromosomes become damaged, and it triggers a DNA damage response. It tells the cell that there's big trouble here, and so you can lose then the ends of those telomeres and all the chromosomes and all the genes that go with them, or sometimes there will be fusion between different chromosomes. So the telomerase is the enzyme that specifically addresses problems with DNA replication to keep the telomeres at an appropriate length to protect the chromosomes.